microbes influence our planet. They drive nutrient cycles, they influence our climate, they break down waste, and many microbial species are still undiscovered. So like every new discovery has the potential of finding something new. Microbes also carry like important genes such as antibiotic resistance, toxin production, etc., and that can affect our ecosystems and affect human health. And so linking the genotype to phenotype is important because it allows us to understand like what these genes actually do rather than just studying genomic information.
So to give a bit quick background of our sampling location, we took a sample from the still water stream from a stormwater management system in front of the Crone Engineered Biosystems building, and we hypothesized that this stream would have more microbial diversity because it was still water.
So our 16S DNA analysis, we got mostly Chloriolis and Bacillus rheophyceae, and the two isolates that we specifically cultured were Pseudomonas otidis and Bacillus thuringiensis. The LB broth and cyclohexamide kept algae and proteobacteria from forming, so that's why we only got those two isolates.
So for a little bit of an overview, the goal of this lab was to research the relationship between genotypes and phenotypes of an isolate from an environmental sample, which ours was the Crohn's building and ours was a water sample. So of the two bacteria that we had in our plate, for Bacillus thuringiensis, we studied UV resistance and hemolysis, and for the Metapseudomonas, we studied motility and antibiotic resistance. So for Bacillus thuringiensis, that's what I studied. It's the most commonly used biological pesticide. It's gram-positive and it's rod-shaped with large clusters as shown on the top left. A couple of the phenotypes that showed up, for motility, it kind of spread all throughout the plate. It showed beta hemolysis and antibiotic resistance to vancomycin and kanamycin. For me specifically, I studied UV resistance using and my gene of interest was the SSPA gene, which is a small acid soluble protein in the alpha family specifically. So for protein structure, because of the protein structure, it has beta sheets and alpha helix folds, which are typical of small acid soluble proteins, and those bind tightly to DNA to prevent UV damage. And then for phylogenetics, a lot of studies with similar species, like Bacillus cereus and Bacillus subtilis, have documented high UV resistance due to various small acid soluble proteins. So that's kind of why I studied that for the Bacillus. My hypothesis was that if you expose the Bacillus thuringiensis to increasing UV times, there will be a log linear decline in colony forming units compared to exposed UV sensitive bacteria. So for my PCR results, I had the lanes 2 and 3, and it kind of showed successful amplification of my gene. And for my experimental design, I exposed Bacillus in liquid cultures to UV times at 0 minutes, 1, 5, and 15, and then measured the colony forming units spread across different serodilutions on plates and compared that to E. coli, which I did the same for those. So for my results, the Bacillus thuringiensis had a 75% survival rate between 0 and 15 minutes. And for the E. coli, which I compared it to, which is UV sensitive, it had a much steeper decline, dropping about 52%. So this kind of supported my hypothesis that Bacillus thuringiensis would exhibit greater UV resistance due to the SSPA gene compared to E. coli, which is shown to be UV sensitive. And then this is a survival curve in percentage. So as you can see, the Bacillus is in orange and the E. coli is the red line, the blue line, and it's much steeper in its decline, which kind of supported my hypothesis. And then these images are across the various UV exposure times. You can't really tell much of a difference in the images just because there's so many colonies, but the Bacillus between 0 and 15 minutes decreased about 50 CFUs, whereas the E. coli decreased about 135 CFUs. So that just further supported my hypothesis. So E. coli showed the expected UV sensitive pattern because there are no small acid soluble genes in E. coli to shield the DNA from UV, whereas in Bacillus, because it had the SSPA gene and a lot of other small acid soluble genes in the genome, those bind to the DNA and change the form, so that helps with the slower decline and UV resistance. So for future directions, something I wanted to study this time, but I didn't get a chance just because there wasn't enough time, was reactivation. So a lot of studies that I saw with the Bacillus cereus and the Bacillus subtilis, they exposed the bacteria to UV and then placed some of the plates in visible light and some of them in the dark, and found that the ones in visible light had some properties of DNA repair. So I would kind of want to study that. And then I'd also want to study exposure to different types of UV. So I exposed mine with UVC, which was artificial UV, but I kind of want to see the differences between natural UV from the sun versus artificial. And then the last thing was longer exposure times. We studied 0, 1, 5, and 15 minutes, but there wasn't like tons of decline in the CFUs. So I would kind of want to see at what exposure time does the bacteria fully, I guess, die.
Sorry.
No, go ahead. You're good.
So I also studied Bacillus thuringiensis, which was gram-positive, shown in the image on the bottom left. It was rod-shaped and large clusters. It had 12.4 megabase pairs. Shown in Isa's slide, it confirmed that there was motility, UV resistance, and antibiotic resistance. And then my specific gene of interest was hemolysin 3. And so this is my protein, hemolysin 3, the image, and then there was documented pore-forming activity in B. cereus when I did some background research, which suggests that there is likely hemolysin 3 functions in Bacillus thuringiensis. There were 9 hemolysin genes present, but they weren't in a cluster, and then they're localized in membrane channels, and they form transmembrane pores for the lysis of red blood cells. And then my hypothesis was Bacillus thuringiensis expressing the hemolysin 3 gene will exhibit beta hemolysis when plated and grown on blood agar, which is characterized as complete hemolysis causing the lysis of erythrocytes. And then here are my PCR results. It was a little difficult to make a conclusive result or interpretation, just because the control didn't really show up, so I'm not sure if they were switched. But then for phenotypic assay results, beta hemolysis was observed, which is shown on the right image, compared to our control, which was B. cereus. But I received the wrong isolate during this lab, so I'm using Isha's B1 results because mine was very similar to Michael and Karen's. So my interpretation of the results, they support my hypothesis. Beta hemolysis was observed, and which aligns with the presence of the hemolysin 3 gene. For future, I would want to rerun the PCR test with multiple hemolysin genes, and then also there was some experimental error just because I think I had Metapseudomonas otitidis during lab 8, so I want to redo the blood agar plates just to confirm that my isolate 2 did have this gene present.
So my isolate was Metapseudomonas otitidis. It had 4 sequences with 6.5 megabase pairs. It was gram stained as negative, you can see that right there. And then there wasn't a lot of linking, so it was pretty rod-shaped. Yeah, they're pretty separated. And then the observed phenotypes that I found were motility, which is what I did, alpha hemolysis, and beta lactamase activity. So I chose motility because there was an operon, or what looked like an operon, in my gene cluster, gene analysis that had a bunch of flagella genes in it, so FLIG, FLGM, FLGN, and then like I think 27 others. And then I specifically chose FLIG because it's part of a protein complex, you can see it right there, that codes for rotational movement. So my hypothesis was that there would be a verifiable presence of flagella on the isolate that I cultured. So this is my PCR results. They were pretty successful. You can see that the control is good, and then that's my isolate sample. So both appeared, so the gene was successfully amplified. And then there's my motility assay. You can see that compared to the positive control, it had a good bit of movement away from the point of inoculation. So I can pretty successfully say that there was motility in the isolate. So based on that, I can say the hypothesis was supported for the most part. I'm not entirely sure if the motility was based on flagella, but because we use a 0.3% agar, I can pretty confidently say that it might have been flagella. And then also the fact that it's Metapseudomonas, which is pretty heavily related to Pseudomonas, which is an order that has a lot of bacteria that have flagella on it. So I can definitely say that there was motility. I can't necessarily say that it was because there was flagella. But if I was able to do another experiment, I could do nano-orange staining. And what that would do, it would stain the flagella on the bacteria, and then I could use microscopy to verify if the bacteria actually has a flagella on it or not.
So my species was also Metapseudomonas competidus. It's gram-negative and rod-shaped, as you can see from the microscopy images, and it also displayed motility, alpha hemolysis, and also antimicrobial activity against the group that had R and clensis. You can see the zone of inhibition there. And the gene of interest that I'm choosing is beta-lactamase activity, specifically metallo-beta-lactamase. So the protein structure was similar to a known beta-lactamase protein. So on the left is a known beta-lactamase structure, and on the right is my protein structure. So it looks pretty similar. And the topology diagram also showed that there was a largely extracellular / periplasmic domains, which makes sense because the beta-lactamase would function in the periplasmic space. I also ran my gene sequence against a known gene sequence of beta-lactamase, and there's a greater than 99% match. And also my functional domain database showed that there's a zinc binding site, which is what a metallo-beta-lactamase would use. It uses zinc to hydrolyze beta-lactam antibiotics, and it also has a metallohydrolyse like MBL fold. So the structure of my protein has this folds that's related to metallo-beta-lactamases. So my hypothesis was that based on the presence of the beta-lactamase gene, I predict my isolate will exhibit antibiotic resistance activity to beta-lactam antibiotics. My PCR results didn't really show banding for the lane with my gene, but my control did have bands. And that can be due to a number of reasons, most likely because my gene sequence had a high GC content, so maybe the temperature didn't really work out, or maybe there wasn't enough template DNA, etc. But for my experiment, I tested antibiotic discs on my bacteria with and without EDTA. And you can see that there's no zone of inhibition around penicillin, but there was a zone of inhibition around gentamicin as well as the combination disc. So what does this mean? This means that my bacteria was resistant to penicillin, but sensitive to gentamicin, and this is characteristic of beta-lactamase activity. But something else that was interesting was that it was also sensitive to the combination disc, but according to literature, my specific subclass of beta-lactamase would not be inhibited by the combination disc inhibitor, but it was. But also the EDTA also killed all of the bacteria on my other plate. So based on these results, I can say that there probably is some type of antibiotic resistance or beta-lactamase activity, but I can't say for certain that it was specifically metallo-beta-lactamase because there was no data from the EDTA to prove that EDTA would inhibit my enzyme. And so in the future, I would use a lesser concentration of EDTA and rerun the experiment, and I would also like to test out more antibiotics or combination discs so I have a larger variety of data.
conclusion, we tested Bacillus thuringiensis and Metapseudomonas otitidis for UV, beta hemolysis, Flagella, and antibiotic resistance. And we found it was UVC resistant. It showed beta hemolysis for Bacillus thuringiensis. And then for Metapseudomonas otitidis, the Flagella alkaline was observed and motility was likely expressed. And then antibiotic resistance was observed, and beta lactamase activity is inconclusive, so we need to do further testing. But studying gaps in microbial research brought us one step closer to understanding the genotypes of unknown bacteria and how they are expressed in the environment. Thank you.
I have a question for Michael and the other flagellar folks. Do you know what kind of flagellum your bacterium makes?
What kind
There are kind of two broad classes of the way flagella are placed on the surface of cells. So all of you working on flagellum might want to think about this. What's the one you might envision in your head? You have a cell, and tell me about what the flagellum looks like.
I mean, like sort of like a tail motor.
And how many?
Probably one.
So there are some like that. I work on Vibrios. They have a single polar flagellum, and it's like a corkscrew, and it goes, when it goes in one direction, that goes straight. When they smell they've run out of food, it goes in the opposite direction, and they just start wiggling. And then when they smell it again, it goes in the same direction, now they spin again. There's another type, another way bacteria present flagella on their surface, and E. coli does this other type. Does anyone know what that is?
I think lateral.
Yeah, it's called peritricious, like perimeter. They have a whole bunch of flagella on the sides, like a whole bunch of tails. And when they go in one direction, they all kind of cluster together like a ponytail, and allows them to swim. Again, when they don't smell food anymore, they go in the opposite direction, they all flop around, and the cell reorients, and then it tightens up and goes. So you might want to just think for each of your bugs, like a picture of, and figure out which of those two categories. Or there might be other interesting ones, but flagellum doesn't just mean one thing. It means a couple things.
Do you know why maybe you only didn't? Because it didn't seem like it even grew on your dish.
I mean, well, ours was probably pathogenic. I mean, Metapseudomonas is like known to be a pathogen, or at least
Oh.
people think it's a pathogen. I'm not entirely sure what Bacillus does.
It's not really. I mean, like, it's just a pesticide, but it's non-toxic to humans. So
Yeah, so a lot of the pathogens were probably selectively outcompeted. Either that, or they were just like killed by the bacteria or the plague conditions. Because yeah, these were like two really small parts of our 16S. Like most of our 16S were like either algae or like alpha potato bacteria. So yeah, I mean, I don't know. I don't have a complete answer to that, but yeah.
Keely?
Since I was sampled from water, I could see how UV resistance would be important with light refracting. Do you guys think that there would be a lot of differences if you sampled in a different section of like the Crohn waterway where there's like standing water, like towards the peak? Do you guys think you would see a lot of differences in the biodiversity in that water compared to the place you sampled at? In terms of like UV resistance, I guess. I don't know if that makes sense.
I don't think so.
You said in standing water?
Like, where that waterway is, if you sampled in a different part, like if you sampled at the top or the bottom of that waterway where it's just standing water, do you think that there would be a lot of differences in UV resistance in the bacteria in those sites compared to where it is where you sampled, like in the channel? Do you think that a lot of
I
them
think
would?
potentially there might be difference in biodiversity. I'm not really sure about UV resistance, just because I feel like Bacillus is a pretty common bacteria, so I feel like it would show up in other parts of the waterway. And if we had our Bacillus thuringiensis I'm not sure about other bacteria.
That's another question then. This is for Isha. How many proteins what was the acronym again?
The small acid soluble protein.
Small acid. So each one works alone? It's
Um,
thought to work alone?
so that's what I was trying to figure out when I was doing research with the Subtilis in this series. Bacteria, in mine specifically, it had, I think, 4 small acid soluble protein gene types. There was the A, O, I, and then H2. And they were all like very spread apart in the genome sequence, so they weren't like clustered near each other. So I don't know if they really work together. In literature, SSP A and B have been studied a lot, but I, O, and H2, there wasn't anything on that, which is why I chose my specific one.
I wonder if there are bacteria that only have one.
Maybe, I don't know. I could see them working together if they're all in the small acid soluble protein kind of cluster. But I'm not sure. I wonder what the differences are and how they work.
And this is a general question for or something to think about for all of you. So other people have said they are working on a homolysin that might just work as a solo protein, and you said you could do things like knock it out. What's another interesting thing you could do that could yield some cool information other than knocking it out? So you got some single protein in your bug of interest that you think does something cool, and you see a phenotype. One is to get rid of it. What's sort of the reverse of that?
You could study it with other hemolysin proteins.
Like introduce something.
So introduce.
Like a gene. Like
Yeah.
Like maybe knock in a different species
Yeah.
instead of knock out.
Yeah, take that gene, PCR it up with its own promoter, or maybe put a strong promoter, put it in E. coli. Now is E. coli suddenly resistant to UV? If that doesn't work, it's hard to interpret. But if that works, that's pretty amazing result, right? That you've taken a non-bacterium that couldn't do this and added back this function. So, I mean, that's a general scheme that is used all the time.
And it's a great way to show that's a single gene phenotype.
Yeah.
Deleting it and not observing the phenotype shows it's essential. But if it doesn't show if it works alone or if it works as a consortium of different proteins.
There's a little caveat to that, which is you can kind of conclude there's no other specific proteins you need to bring over. It could be that E. coli has another thing that like all cells have that it can also work with, right? But it's really nice evidence that this alone, and you literally just brought one thing alone into E. coli, and now E. coli is doing it. It's pretty cool.
The caveat is that one is gram positive, one's gram negative.
So maybe you move it into exactly. Or move it into a closely related organism, like a common bacillus strain or something. Put it in, so E. coli is like the model gram negative. There's model gram positives too. But this idea of adding function is the reverse of deleting a function, right?
Can we go to carry your PBTA?
Yeah. Like the plates.
PBTA plates, antibiotic resistance. So something caught my attention on the top right. There's a disc, right?
Yeah.
And on top of it, there's growth. And at the bottom, there is no growth.
Yeah, it's just refraction. It's just
Is that light or is that cells? I can't.
It's just a light.
It's refraction, sorry, when I took the picture.
The yellow,
My bad.
the yellow zone is light coming from the ceiling, sort of.
No, on the way I took
On the gent? Up in the gent, on the right.
the picture, when I took the picture, it's
Because it looks like it goes around the disc, right?
It's like a
But you're also changing the auger angle, so maybe it's not refracting the same way anymore.
Make sure you know.
Are those plates still here?
Yeah.
The plates are still here too. Yeah. Yeah, take another picture.
I saw a lot of photos here. Because this was on, you can just take it off.
Yes. Yeah, you can do that if we have time before we leave. Take a picture if you want another one.
25 minutes.
Yeah, it's 25.
Okay, great. Thank you.